Hello, everyone. I'm Joshua Stein, Project Manager at the International Wheat Genome Sequencing Consortium, and it is my pleasure today to serve as moderator for today's monthly webinar series. So welcome, everyone. Today, we will be joined by Andrea Gonzalez, who will be speaking about the Aegilops tauschii pangenome and diversity within. Let's see, we have, okay. I think we'll continue. We have about 65 attendees so far. Before we continue on to Andrea's presentation, I'd like to give you a quick overview of the IWGSC. We are an international nonprofit consortium with more than 3,400 members belonging to over 900 institutions and companies, and they're engaged with us from 72 countries. None of this would be possible, including our monthly webinar series, the IWGSC annual workshops, and coordination activities related to genome resources without our 9 sponsors. And so I'd like to take this time to thank our sponsors, Arbor Biosciences, BASF, CIMMYT, Florimond Decree, INRA-E, Illumina, Kansas Wheat, RAGT, and Syngenta. So thank you to our sponsors. As most of you know, the IWGSC was instrumental in the production and release of the first bred wheat reference sequence back in 2018. Our mission since that time has been to develop additional tools and resources to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. This has included updated versions of the reference genome, updates and refinements to gene annotation, especially through a manual curation effort, and functional curation of gene families, as well as contributions to the study of landrace diversity. So just to give a quick overview of the dashboard, you can ask questions at any time throughout the presentation using the question icon. And at the end of the presentation, Andrea will have an opportunity to answer those questions. If there are too many questions and we run out of time, Andrea has agreed to respond to all the questions in writing, and we will provide a link to those as well as to the recorded video. And this will be posted both at the IWGSC YouTube channel, which you can see at the top of this slide, as well as at the IWGSC website, which is wheatgenome.org. If you experience a technical issue, please use the chat. Our technicians will be monitoring the chat channel and can respond to any technical issues. As well, please monitor the chat in case our organizers send out messages such as links to papers and that kinds of thing. Already you can download this introduction as well as Andrea's presentation as a handout in PDF format, so please go ahead and do that if you'd like. Oops. And with that, it is my pleasure to welcome Andrea Gonzalez. She is a PhD student and candidate at the laboratory of Dr. Brandy Wulff at the King Abdullah University of Science and Technology. And today she will be talking to us about the Aegilops tauschii pangenome, a tool for harnessing wild wheat genetic diversity. So thank you, Andrea, for sharing your work today, and thank you all of our attendees for attending today. Oh, and I just want to mention our next webinar will be in October, so please stay tuned for information sent out about that. Okay, thank you, Andrea. I'll let you take over.
Okay, thank you, Josh, for the introduction. And I want to thank everyone who is attending this webinar. Yes, today I will present, and I'm not seeing my screen. Oh, that's not it, right? Let me minimize. Are you, yes. Okay, I will present the results on the Pangenome of the wild wheat Aegilops tauschii. So this is a genomic resource that has been generated under the aegis of the Open Wild Wheat Consortium, and it's being coordinated by the Wulff Lab, which I am part of, at the King Abdullah University of Science and Technology in Saudi Arabia. So I want to begin by introducing the Open Wild Wheat Consortium to those who may not have heard of it yet. This is an international research effort in Triticum, and the main aim is to generate wild wheat resources that can contribute to breeding, genomics, genetics, pathology, and germplasm, and all of this looking back to wild wheat that can contribute to bread wheat improvement. So it's a collaboration of researchers from across 15 countries and 26 research institutes and universities that are listed here. So currently the leading effort of Open Wild Wheat is focused on Aegilops tauschii. So tauschii is the D-genome donor of hexaploid wheat. It's a diploid, and its genome size is around 4 gigabases, which is 4 times less than hexaploid wheat, which makes it a suitable, more convenient study organism, especially if we want to assemble genomes. It's slightly less complex. And Aegilops tauschii has a high genetic diversity that was lost or not captured entirely when tauschii hybridized with the tetraploid AB genome donor to give rise to hexaploid wheat. Only a fraction of this large diversity was captured, and then breeding and selection further created a genetic bottleneck, whereas now we look back to tauschii and other wild wheats to capture this genetic diversity in terms of traits of strategic interest, such as pest resistance, disease resistance to rust, powdery mildew, blotch, and also plant architecture traits that are important, such as trichome density, and other traits such as tolerance to, sorry, the light one. Sorry about that. So tolerance to heat, drought, salinity. So Open Wild Wheat began in 2017. It's phase 1. And in 2021, it ended with the publication in Nature Biotechnology on population genomics of tauschii. And so there were several key findings or key results in this first phase that I'm going to highlight now in terms of genomic resources and in terms of, well, results, actual findings. So the first one was that diversity panel of 242 non-redundant extensions of Aegilops tauschii was consolidated, and this panel spanned from Turkey to Northwest China. In addition, in this panel, a third lineage of tauschii was discovered. So Aegilops tauschii has a population structure divided into lineages, and lineage 3 was discovered, restricted geographically to Georgia and represented in the panel by 5 non-redundant accessions, which are shown in the phylogeny. This is a phylogeny of lineage 1 accessions of the panel, shown in yellow, lineage 2 accessions shown in blue, and lineage 3 accessions shown in orange. And in green, we see the D-genome of hexaploid wheat of land races. Another key finding was that it was estimated that only 25% of the diversity of Aegilops tauschii actually hybridized or was represented in the wheat land races. And a panel of synthetic hexaploid wheat was generated that captured or restored that genetic diversity to 70%, including diversity from the 3 lineages, lineage 1, lineage 2, and lineage 3. The diversity panel of 242 accessions was also used to to sequence these lines and phenotype these lines for several traits of interest, such as trichome number, spikelet number, powdery mildew, resistance to powdery mildew, and resistance to wheat chromite. And these traits show variation, a continuous variation across the panel, and this variation was important, was used in association genetics to identify LD blocks associated with these traits, and potentially so that genes can be cloned from these LD blocks, or identify these genes that control these traits. So this resource was very important because it set the groundwork for a genomic resource and a panel that can facilitate gene cloning, association genetics, and other areas of study in wild wheat. But it did have its limitations. So one of the limitations of this resource is that while the panel included 242 accessions, remember we have a population structure. So actually in each lineage, there was just a little over 100 lines or accessions that were represented in lineage 1 and lineage 2. And when lineage 3, it was just discovered, so there was only 5. So that poses a limitation or the power of detecting rare alleles that are controlling traits of interest. And so one solution is, well, let's increase the size of the diversity panel so that we can improve association genetics. And also related to association genetics, a limitation at the time when phase 1 was done, when it was published, there was only one reference genome published, and that was for AL878. And there's a limitation in the reference genomes that you use for association genetics. This is an example of a GWAS plot where a peak was detected in chromosome, in the short arm of chromosome 70, but this is using the AL878 reference genome. But when they went into the region, into the LD block, they found out that really the gene that could be most like that is associated was not present in the reference assembly. And this is a limiting factor. So what's the solution? To generate a pangenome, to generate high quality assemblies that can represent the diversity of the panel and that can increase the resolution of association genetics and other studies. So now I'm going to go into the results of Open Wild Wheat Phase 2, and this is what I've been working on my PhD thesis. It started in 2021, and now we're 2 years in, and well, the first goal was to expand the diversity panel. So currently we have a panel of 675 accessions that we have seed for here in KAUST. This is a work that's also, well, in our lab, Catherine Gardner, Ana Belén, and Chai, they've been working on as well. And we started from the collection that was in the OWWC, the first phase, which was 242 accessions. And we've added accessions from different collections of germplasm banks and also private collections or donations from, for example, the Tajikistan National Academy of Sciences, from Azerbaijan, and Quaid-i-Azam University. So all of these accessions add up to 675. We've sequenced most of them. So we've sequenced 621 out of the 675, and we're still looking to receive new collections. So right now, a possible future addition is an Iranian collection from Professor Ali Merabi with over 50 accessions. Now this is a map of the distribution of the current diversity panel. So we still go from Turkey, but now we've expanded more into China to include accessions from most Eastern China. And we've increased the number of accessions of lineage 1 and lineage 2. As I mentioned, we've been able to sequence 621 accessions out of 675, and this was done with Illumina whole genome shotgun sequencing at an average of 7.5 coverage. And we've also included for our genomic study public sequencing data that's been available for 324 accessions, and these were published in recent papers in Nature Plants. So that gives us a total of 945 accessions that we're studying at the genomic level that we have sequencing data for. And of course, we expect a certain amount of redundancy, of genetic redundancy in the panel. So we analyzed the amount of genetic redundancy and found that 511 accessions we considered as non-redundant, and found on a percentage of the genetic redundancy around 40% for lineage 1 and 2. We managed to increase the number of non-redundant accessions in lineage 3 by 1, so we went from 5 to 6. It's still a rare lineage to detect. And this clustered heat map shows the 945 accessions. Blue is lineage 2 accessions, yellow is lineage 1 accessions, and here are the lineage 3 accessions. As it has been reported that lineage 2 is much more genetically diverse than lineage 1, which is now we see it in the plot. There are groups within lineage 2, but we also see some groups within clusters within lineage 1. Well, a diversity panel is only as informative as if we have high quality genomes that we can anchor it to, that we can study it. So the next step, having this large diversity panel, was to generate the pangenome. And for this, we selected 44 accessions out of the diversity panel. These accessions were selected based on 2 criteria. One was strategic traits of interest. So for this, we reached out to the consortium and we asked, please nominate accessions that you're working on, that you have a trait that you're pursuing, that you want to clone a gene. And we gathered quite a list of accessions that later we had to narrow down, but these accessions were nominated based on disease and pest resistance, salinity, tolerance, yields, other traits of interest. And then the second criterion was that we had to, of course, a pangenome represents the diversity of a diversity panel. So we selected then the accessions based on the phylogeny and to try to at least represent the major clades within each of the lineages. And this is the distribution of the pangenome lines. Based on findings that lineage 2 is more genetically diverse than lineage 1, there is a higher representation of lineage 2 lines in the pangenome compared to lineage 1, and a single line of lineage 3 because it's found that they're very genetically similar. There are pangenome accessions that are not shown in the phylogeny, and I've listed them here. So for the pangenome accessions, we sequenced them with PacBio. So we generated PacBio hi-fi reads for them and assembled primary assemblies from these. So I'm going to show here just some basic statistics of the sequencing data. So in terms of coverage, we sequenced them at an average of 23x coverage, but anywhere between 18 to 28. And this large range of coverage is was due to a large range of contig N50s that we also found. So the assemblies that were sequenced at a much higher coverage was in an attempt to increase the N50. But we found that we didn't find a correlation really between coverage and N50. But we did, this plot shows N50 length and coverage, but we did find that there was a difference in the contiguity of the assemblies depending on which lineage the lines belong to. So here we see a lineage 1 accessions in blue, lineage 2 accessions in orange, and lineage 3 accession in green. And lineage 2 accessions were easier to assemble than lineage 1 accessions. Even if they were sequenced at a much higher coverage, even close to 30x coverage, the N50 wouldn't exceed 30 megabases. Whereas with just below 22x coverage, we achieved an N50 of 90 megabases for one of the lines or one of the accessions of lineage 2. So we believe that this could be due to a biological reason, a biological difference between lineage 1 and 2 accessions that impact the contiguity of the assemblies. I'm just highlighting here the best assemblies, and I want to highlight TA1675 because that became a very important accession in the Pangenome. And we're lucky because we selected it arbitrarily just because we had more seed for it, but we were lucky to get a good line to assemble. We also evaluated the Pangenome assemblies in terms of accuracy. So for all of the Pangenome lines, we also have Illumina data for them. And so we assessed the base call accuracy, which was really high, an average of 46 on the Phred score, which is equivalent to over 99.99%. And then we also assessed the correctness with a Busco completeness of over 98, on average 98.4. So we were satisfied with these assemblies, but can also remembering that Aegilops tauschii has a population structure of lineages, thought it's important if we generate chromosome scale assemblies for each lineage, and that way we could anchor the pangenome to a lineage level reference. And this because this can improve the resolution when we're doing SNP calling, structural variant calling, because the reference that you use for these is very, very important, and it could be a limiting step if you're not capturing really the diversity that's there. You could be missing out on due to presence-absence variations. So to generate these chromosome level assemblies per lineage, we increased the PacBio Hi-Fi reads coverage from 67 to 97x coverage, depending on the line. And we sequenced, we added Highsea Illumina reads, 300 million reads. After this, we generated phased assemblies and scaffolded these assemblies to the chromosome scale. So here I'm showing the 3 lines that we selected to be the reference assemblies for each lineage. So lineage 1 is TA10171, lineage 2 is TA1675, which I mentioned earlier, and lineage 3, which is our only lineage 3 line, 2576. And here's the comparison. What I want to highlight here is they're comparable to the existing NCBI RefSec assembly, which is AL878, in terms of the assembled length, and in terms of the unplaced scaffolds, which is actually much less than the current reference. So these are assemblies that are at reference level. This is to go a little bit more into detail about the Highsea scaffolding of the assemblies. So I mentioned that we had different coverages. The L1 line was sequenced to 77x, the L2 line to 97x, and the L3 line to 67x. And so we started the starting point for the Highsea scaffolding was based on assemblies with a different contig N50. And here I highlight again that the contig N50 was drastically different between lineage 2 and lineage 1, whereas even at 77x coverage, we didn't exceed over 50, it was 53 megabases, compared to lineage 2 at an impressive 221 megabase contig N50. These are the Highsea contact plots, where each of the blue squares shows the chromosomes, the 7 chromosomes, and the green squares show the contigs that were scaffolded into chromosomes. But the next figure was based on these, and I just want to highlight here that we did see an interlineage variation in number and the location of gaps. So for example, I want to highlight the lineage 2 accession, which was really impressive line to assemble, because we were able to first assemble chromosome 70 in a single contig, and also assemble the centromeric region, which is highlighted by, shown here by the asterisk. Most of them, for most of the chromosomes, it was assembled in a single contig. And so that explains the very, very high N50 value and the contiguity of this assembly. And in contrast, L1, the centromeric regions were not assembled into a single contig. They were very fragmented. as well as the telomeric regions. And L3 is kind of an intermediate between both of them. Sometimes we did assemble the centromeric region, sometimes not. It's not as fragmented. But yes, the number of gaps in total is low, and mostly for this accession. So here, this is an overview of the pangenome then, the 44 lines that we've sequenced. Again, here are the L2 lines. We see a diversity, or yes, there's genetic variation among the L2 lines that were chosen, indicated here by the groups that we see. Then we have the L1 lines, less variation, but we also see a diversity, and then our single L3 line. And then this is the Kamer accumulation curve. These Kamers were generated from the genome assemblies, and this is to show that the pangenome, as we add a genome, we do add diversity to the pangenome, but we still haven't plateaued, so there's still some diversity to sample. However, it's representative of the panel. So with this, I'm wrapping up what's presenting the diversity panel, presenting the results of the pangenome, and now for the rest of the talk, I want to say, what are we doing with this? What are the applications of the diversity panel and of the pangenome? So it's a resource that's meant to facilitate gene discovery and annotation, aid in evolutionary studies, especially in association genetics, which is what my lab focuses on a lot, and my work focuses on a lot, detection of structural and regulatory variants, gene cloning, genome engineering, conventional breeding, and many other applications that I don't mention here. And I'm going to give a couple examples of what we've been working on, just quick examples to show how the pangenome has improved these studies. So one of them is in association genetics. And here, I'm going to show how just having a high quality reference genome assembly can improve association genetics, especially because it reduces the noise that we see. So this is a Kamer GWAS plot showing the associated Kamers for resistance to stem rust, to Buccina graminis, in an accession in TA1662. So this plot is generated using a low quality reference assembly. It's a Contig N50 of 196 kilobases. This was generated with Illumina data. And yes, we see the peak corresponding to a known gene, this SRT1662 that's present, but there's some noise here that you can interpret, is this a peak? Is this also relevant? But once we add a high quality reference genome, the noise disappears and it's much clearer what we're pursuing. So these are not seen anymore in a high quality assembly with a high Contig N50. And the same goes for another trait, which is trichome number in the same accession. So here we have the low quality assembly. We have a lot of noise here. Is this a peak? Is this a peak? Now, when we use a high quality reference, these peaks disappear. So it's much clearer that this is the main peak that we can pursue, which was in the other plot. This one as well. These disappear, and then we detect another peak that would be worth pursuing that wasn't exactly visible here because it was masked by this one. Another application, another example that I'm going to touch on is gene discovery and annotation and gene cloning. So here, this was an exercise that we did. Okay, it's based on previous work. It was published in 2013 by Olson and collaborators. So they mapped SRTA1662 by introgression into wheat. And so SRTA1662 was mapped to chromosome 1D, co-localized with a known stem rust resistance genes, which is SR33, that confers resistance to UG99. And they mapped this segment and introgressed it into wheat and found that it also conferred resistance to UG99. But in the OWWC paper that was published in Nature Biotechnology in 2022, the gene was cloned. So here it wasn't cloned, it was just mapped to the same location as SR33. The gene was cloned and it was found that it does not confer resistance to stem rust isolate UG99. So the hypothesis is that the introgress segment containing SRTA1662 must contain another resistance gene that is conferring that resistance to UG99. So the exercise that we did is now having a high quality assembly for TA1662, which is the accession where this gene was cloned from. We mapped the markers that were flanking the gene. It mapped to a single contig of 13.76 megabases. Then we annotated, predicted the NLR loci in that contig, finding that they were predicted in this region. And then we predicted the genes within those NLR loci, found 9 possible genes. One of them was SRTA1662. Then we have expression data for this line, and then comparing with the expression data, only 4 of these 9 genes are expressed. One of them has a stop codon, a premature stop codon, which left 2 genes that can be potential stem rust candidate genes that are conferring the resistance to UG99 and are in this interval in addition to SRTA1662. So now we are functionally validating these genes to see if they actually are involved in this resistance. And this example also allowed another opportunity there was a question of, is SRTA1662 and SR33, are they alleles or are they paralogs? Because they map to the same location on chromosome 1D. And at the time, yes, SR33 confers resistance to UG99, but SRTA1662 doesn't. But are they different genes or just alleles? So there are several assumptions that they could be different genes. One is they have different specificities, as I mentioned, but that could still be because they're alleles. What we also found is they have low protein sequence identity. So it's 83% sequence identity between them. Whereas genes, for example, SR33, different genes of SR33 in different lines have an identity above 96%. But what really confirmed or led us to believe that they are paralogs and not alleles is that SRTA1662 and SR33 are in cis. So they're localized in cis. And this was because we mapped these genes to 4 assemblies of the pangenome. One of them is TA1662. The other ones are assemblies that were reported to contain SR33. So this shows a fragment of chromosome 1D for each of the assemblies. This star shows SRTA1662 gene. The other star shows SR33 gene in these lines. And we found one line that contains both genes. So that is the evidence that these are in cis. They are localized in the same chromosome and in the same line. So this is evidence that we can now give a real stem rust resistance gene designation to SRTA1662. And the last example that I will touch on is another example of gene discovery and annotation that also includes association genetics and gene cloning. And this is related to leaf trichomes. So this is a trait of interest that we're pursuing in our lab. We're dissecting leaf trichome regulation. We're using this trait because it's been used to characterize wheat. It's a trait of easy observation. So leaf trichomes can be phenotyped as early as 2 weeks. In seedlings of 2 weeks old. It has high heritability and it's been linked to pest resistance and abiotic stress. And in the previous paper, in the panel of 242 lines, there's a continuous variation in this trait where you can see anything from lines that have 0 leaf trichomes to intermediate number of leaf trichomes to very high. And in the previous paper, an LD block in chromosome, in the long arm of chromosome 4D, was identified as potentially related to this trait. But with a large panel, and comes challenges. So we have a challenge to face. It's how do we phenotype trichomes in a way that's consistent, reproducible, that can be used to accurately measure leaf trichomes. So before, just photographing was difficult. What is the unit of measurement? How do we normalize the scale at which we take the picture, the depth, the lighting? It was an issue. So in our lab, Catherine Gardner and collaborator Martin Clint developed the Trichochem, and it allows high throughput phenotyping of trichomes, taking images that are all at the same depth with the same lighting, and this allows phenotyping panels of over, let's say 675 lines quickly and in a very reproducible way. And the importance of this, or the main idea of this, is once you have all this data, how do you process it? Do you manually count the trichomes? And that's not really feasible if you have a large panel. So with this we also want to do an automation process for trichome counting. So using artificial intelligence and in association with Dia technologies, using these images, that these are the images that the Trichochem generates, that are 2 by 2 and they're at the same scale, counting trichomes and detecting them through AI. Sorry, this is the TiaTech platform, and it detects them and counts them based on training with different images. So now we've achieved accuracies of a little over 85 to 90%, and we're still working on improving the algorithm, but this is greatly facilitates high throughput, large scale phenotyping, especially on a panel of this size and other panels potentially. So now that we have the trichome phenotyping problem solved, now the next step in which we're going at is dissecting leaf trichome regulation. So it's studying the variation, and for this, the Pangenome provides a very good example of how just having a different reference assemblies, we also see not only phenotypic variation, but a genetic variation or evidence that this trait is controlled by different genes. So this is just some examples of a line that has 0 to very low trichomes. We see a certain number of peaks, but when we see lines that have an intermediate number of trichomes, other peaks start to appear that could be potentially controlling the trait. Some LD blocks are now positively correlated, negatively correlated. And then when we look at very high trichome lines and we have a reference assembly for that one, other peaks start to show up. So that shows the value of having this pangenome where we could really, really study the trait across the panel. So that was the last example. And now to wrap up, just some take-home messages. So we have an Aegilops tauschii diversity panel, one of the largest wild wheat panels available right now, of 511 non-redundant sequenced accessions. A tauschii pangenome of 44 high-quality genome assemblies. 3 chromosome scale reference assemblies, 1 for each lineage, and this serves to anchor the pangenome, and the pangenome serves to anchor the diversity panel. And all of these resources, they're for enabling high-resolution genetic association studies, gene cloning, detecting structural variants, genome engineering, and many other applications that I haven't even touched on here, but that will be high value to the Open Wild Wheat Consortium and to just the wheat community in general. So with this, I want to just acknowledge the Wulff Lab, my group, Brande Wulff, our PI, our group leader, and everybody who's contributed in any way to the project. And especially Chai, because she's not in the group anymore, but she was very, very pivotal to the first stages of the project, collecting all of the seeds and extracting DNA, RNA. Also, I want to acknowledge Simon Kratinger's lab and Jesse Poland's lab here at KAUST, and PhD students, postdocs that work with their lab that have really contributed to this work. Also, Qing Gao's group, he's a PI also here at KAUST, and Salim and Adil, who have contributed to generating pipelines and code for processing all of this data, especially for K-mer matrices and for the association genetics. And Luis Rivera for providing technical support to all of this. The CDA, the Center for Desert Agriculture here at KAUST, Taya Technology and its Chief Executive Officer, Justine, who's helping us with the AI detection for trichomes, the John Innes Center, and the Open Wild Wheat Consortium, which is, like I mentioned, 20 researchers from 26 institutes, and in particular, these institutes that have donated seed to consolidate the panel. And with this, I want to thank you for attending the webinar, for listening, and I am open to your questions. Thank you very much.
Okay. I don't know if people see me, but thank you, Andrea, for a really interesting and terrific talk. I see a number of questions are rolling in. We have about 5 questions so far, and they continue to roll in. The first question was asked very early in the talk, and I'm not sure how to interpret the question, and maybe it needs to be rephrased, but it says, why line A or why linea? So I'm not clear on that. Andrea, if you understand that question, you can go ahead. Otherwise, I urge the asker to provide a little more context to the question.
Yes, I don't understand either. Why line A?
Yeah, or linea. Okay, so why don't we move on? The next question is, did you use R Passset for data?
R Passset is,
or R packet, I guess.
Oh, R packages for, and no, I don't use R. So no, not for this. For this, we've mainly to generate the non-redundancy analysis, all the analyses on the panel, we use K-mers. So for this, these are pipelines and code that we've developed in our lab. They're in-house codes.
Okay. Here's a question. It says, great work with the genome assemblies. This question refers to the K-mer GWAS. Why does the vertical axis always start at 6 rather than 0?
Okay, that's a cutoff that has been found. It's more to reduce noise because it starts off at 6. Just anything below that is not considered significantly associated. And actually, it starts at 6, but the real cutoff, there's a dashed line at 9.2. So to call a peak, we call a peak at everything above 9.2. Okay. And this was just determined. It was determined in the previous OWWC paper based on just positive controls that's what something really associated is worth calling.
Okay. The next question, do trichomes contain insect feedants such as polyphenols?
I don't know, but an interesting field of study would be to do metabolomics.
Perhaps anti-insect, I think was meant.
Yeah, probably. Yes, because it's true, because it's associated with pest resistance and abiotic stress, but I don't know that. but it would be interesting to pursue metabolomics on trichomes because trichomes have been associated with health issues in humans. And one thing could be with respiratory problems. And the question is, well, what do they contain? What is it that's creating these problems? So that's
yeah, worthwhile area to pursue. Okay, here's another question. Thanks for the nice talk. In your opinion, what are the major barriers to look deep into Aegilops tauschii genome at this point? And what is the way forward using this D-genome for wheat improvement in terms of techniques and resources?
okay. That's a good question. Well, let's see. Major barriers that we're facing right now. Well, assembling the genomes was good with tauschii. We're excited about that. But now that when we get to these functional candidates that we want to validate, I know working on transforming tauschii or diploids in general, is not as straightforward as, for example, transformation in wheat that's running now and it's achievable. Transforming a diploid for some reason is much more difficult. And so doing CRISPR-Cas knockouts in tauschii is not so straightforward. So that would be one of the major barriers that we're facing right now.
What about the generation of, say, synthetic hexaploid wheat lines with new D-genome donors? Is that something that your consortium is working toward or interested in?
Well, I don't know. Our lab isn't, but since it's been done before and it was done in the OWWC paper, and let's say there's a protocol established, I guess it would just be worthwhile getting, since we've sampled a much higher diversity, guess it's something that could be done, but I don't know if somebody's going to pursue it.
Okay, so I think this gets back to the original question. Why is lineage 3 underrepresented among the 44 lines selected? I believe you touched on that in your talk, but maybe you can just rephrase that.
Yes, well, lineage 3 is a new lineage that was discovered in the previous paper. And it's geographically restricted to Georgia. So let's say it's more of a sampling, perhaps a sampling issue, because the lines have been collected from Turkey, from the geographical distribution range that I'd mentioned. And perhaps if we do more sampling expeditions to Georgia, we could potentially increase the size of the L3 panel or the L3 lines, but it hasn't been done. So now, I mean, the reason it's underrepresented is because we're taking lines that are available from germplasm banks, and these lines have been collected in 1940s from several expeditions, historically from 1900s to that. So we're just limited to what is available and has been collected.
Right. Okay. Okay, and this this is by an attendee who missed the beginning of the webinar, but is curious how the GWAS for trait discovery is done using K-mer counts.
Okay, good. That's a good question. Well, first of all, why do we use K-mers and not SNPs? Because traditional GWAS uses SNPs. So there's a lot of bias. SNPs depend on the reference genome that you're calling them against, which is the reason why we use K-mers. K-mers are reference free. And the question is how we do it or why we do it?
I guess it's how. That was the question. Yeah.
All right. So the how is we take the whole diversity panel has been sequenced. So we take Illumina data, the sequencing data from that panel, and we generate K-mer sets for each of the lines. So the K-mers for all of these lines. And then we compare those K-mers across the entire panel and generate a matrix of K-mer presence absence variation. And that presence absence variation is fed into the GWAS pipeline. and is used to correlate it with the phenotype. If whoever asked this question and anybody else, if they want more detail about how we do a KAME or GWAS, it's referenced in the OWWC paper and in another publication that I believe I shared. It's the Agrenseq. That's where the original KAME or GWAS was developed. So I believe I shared that reference in the webinar information. So whoever's interested could go back to the paper and see how it's done.
Okay. Here's a question that I'm going to sort of combine a couple of questions about the availability of these resources, the assemblies, and also whether genome annotation has been carried out on these genomes, and when these might be available, if so.
Okay, yes. So if you visit the openwildwheat.org page, within the next few days, we will make this data available because it was a pledge that we made. This data will be available under a Toronto agreement, the Pangenome assemblies. They can be downloaded by contacting us. So that is pre-publication access. We don't have genome annotation for these yet, but it's a work in progress. So we're generating the data for that, and they will be annotated within this year, hopefully. Because for the, so yeah, right now, for the work that we do mostly with GWAS, it wasn't a priority, let's say, but it is something that will complete the Pangenome. So yes, please be on the lookout for the access to the data.
Okay. Here is a question. Some reports say that L3 is a hybrid between L1 and L2. Based on your results, do you believe that this is true or correct?
Actually, it was shown in the first Open Wild Wheat paper, so the one that I referenced, the population genomics analysis. It was shown that L3 is not a hybrid between lineage 1 and 2. That's actually, it's a separate lineage. And this was based on Kamer-based structure analyses, where, I don't have a figure, but at K, anything above K4, K5, K3, it was separated. The lineage 3 accessions are not a hybrid. And the other way, I'm gonna go, another support for this is seen in the phylogeny. I have to go back a lot. In the phylogeny that was generated in the OWWC paper, they generated a recombinant inbred line on purpose to test this. So there's one line here, which is this one, symbolized by R. This is a line that is a hybrid between lineage 1 and 2. And you can see it does not group with lineage 3, and it groups outside of the lineages. It groups with lineage 1, but it's not, if these were hybrids, you would see it in the phylogeny. So they proved by structure analyses and Kamer-based phylogeny that it is not a hybrid.
Thank you very much. That's very clear.
Okay,
Now,
now I don't.
Oh, go ahead. Hello?
I don't see you anymore, but.
You don't see me? I'm sorry.
No. Let me go back. That's okay.
Okay, well, as long as you can still hear me, we have about 5 minutes. I still have a few more questions we can try to get through. Here's one. Was the AUSH 18911 accession, was that the only one that had both the R33 and the TA1662 genes, or do you know of any other accessions that also have both?
Okay, yes, well, this was, it was an exercise. Why those 4? I've only analyzed them on those 4 accessions, and it was because the reported SR33 gene sequences were for those 3 accessions. AUS 18911, 18913, and CPI, the other one. So we were lucky to find that in that accession. It was unexpected, but we haven't looked at it in the entire pangenome, but there probably are other accessions that contain both, and that's something that we're also going to screen against the other lines.
Okay, yeah. Okay, here's a question. Very interesting work. Thank you very much. There are some reports that tauschii genes would not express at the hexaploid level. What is your plan to link directly with hexaploid wheat? I think the question was cut off. So I guess the initial question is, is it thought that the tauschii genome is somehow suppressed within the hexaploid context?
Yes, I don't know. I can't say that what are the strategies, because it's not an area that I have expertise in, but I do know that that does happen when you try to express a gene or introgress it back into wheat. Somehow it does not get expressed. So yes, the hypothesis is exactly what the question says, but I don't really have much more else to add to it.
Okay. Here's another question concerning disease resistance genes. The attendee wants to thank you and tell you what a great job you did. In addition, the person says, I really wanted to know target major wheat diseases you considered to study for disease resistance traits.
Well, our lab focuses mainly on stem rust. So that is one that we're actively researching. But stripe rust, leaf rust, all of these are. Oh, we also study leaf rust. I mean, all of these are major diseases that cause major loss to wheat. So, I mean, I would say any of the rust, really.
Okay. I think we have one final question here. Oh no, a couple more questions. Did you try GWAS for L1 and L2 independently?
Actually, we are, yes. We have, I don't have the results on that, and it's very preliminary, but we have been running GWAS for only L1s. The thing is, there's the L1, since they're not as genetically diverse, there's a redundancy there that makes GWAS challenging because we're seeing a lot of noise. So I guess there's a lot of tuning to be done if you're running it just on a single lineage, especially for L1. So we don't have that down yet, because when we run a GWAS, of course, there's a population structure analysis, a PCA that's done in order to detect the underlying population structure and to mask k-mers that are not associated with the trait, but more with the population structure. And that's proved to be challenging when we're running it just for that lineage in particular. Maybe it's the trait that we're analyzing, but we have faced this challenge.
Okay, all right, so we have about 1 minute left. I think there are maybe 2 remaining questions that could be answered in writing. One is about specific phenotypic traits within lineage 3 that might differentiate it from other groups or lineages. So you may be able to elaborate on that in writing. And then another question about why lineage 3 is so closely related to lineage 2 phylogenetically. Given that we're running short on time, I just want to once again thank all of the attendees for joining us today, and I want to thank Andrea for a very interesting and informative talk and for agreeing to do the talk today. Andrea, if you'd like to make any final remarks, you can.
Yes. Hold on, I'm gonna stop sharing because I don't. Now I see. Good. No, thank you. Thank you for the invitation, Josh, and to Isabel and to Rolanda for arranging all of this, and to the IWGSC. Yes, I'm honored to to have presented the webinar and to everyone who joined and their interest, and the questions were great. So whatever, I will be sure to get back to anyone and elaborate on maybe some answers that weren't that well, but yeah, definitely.
Okay, terrific. All right, well, thank you again, everybody. We'll see you in October.
Okay, thank you.
Okay,
Bye.
all right, bye-bye.